Okay, so for our lecture session today, we are going to continue with chapter 3, which is still on the chapter of plasma membrane transport. Okay, so on Tuesday's lecture, we have looked at until the presence of cholesterol. Okay, the presence of cholesterol in the plasma membrane. So by now you should be able to explain the structure of plasma membrane. Okay, the components of the plasma membrane, the fluid mosaic model. Why is it called as fluid mosaic model? Okay, and then for example, okay, uh, in the plasma membrane composed of mainly the main structure of it is composed of phospholipid bilayer, and then you have other component that is embedded in the membrane where you have the integral protein, peripheral protein, you have the cholesterol, you have the glycoprotein, you have the glycolipids. Okay, so all that component you have to know how to draw it, you have to know how to label them, you have to know the function of each one. Okay, so basically for the protein that is embedded in the membrane, we are going to look at in detail during this lecture session. Okay, so all right. So in this session, we are going to look at subtopic 3.3, which is membrane proteins and their function. Um, okay, so it says here proteins determines most of the membrane function. Okay, most of the membrane's function. Why it says like that? So the presence of the protein, for example, it allows molecules or ions, certain molecules or ion to pass through the membrane. So this ion will have their function in the cell. For example, in the neurons of cell, in the neuron cell. Okay, the movement of ions, sodium and chloride ion, for example, in and out of the cell through channel protein. Okay, uh, this is to generate impulse. Okay, for example, impulse. So the generation of impulse by the neuron is due to the movement of ions in and out of the cell through the proteins that is embedded in the membrane. Okay, so other functions of the protein, for example, it can function as a receptor for hormones. Okay, so hormones that are made up of proteins, okay, they will bind to the protein that is embedded in the membrane. Okay, so hormone that is made up of protein, they cannot enter into the cell, they will just bind to the membrane protein, and then the protein will relay information into the cell, so that the cell can catalyze certain reaction. Okay, so those are the example of how proteins will determine the membrane's function. Okay, so as you know, there are two types of membrane proteins. The first one here, you have the peripheral proteins. Okay, so it says here bound to the surface of the membrane. So you just add to your note. Okay, surface of the membrane that is facing the cytoplasmic site. So remember that, facing the cytoplasmic site. If you were to draw the peripheral protein, you draw the protein near the surface of the membrane, at the surface of the membrane, but facing outside, facing the extracellular fluid side. So your peripheral protein will be wrong. Okay, peripheral protein, it is at the surface of membrane facing the cytoplasm. Okay, so this peripheral protein can attach to the cytoskeleton in the cytoplasmic site. Okay, this is to enhance the or to maintain, okay, the membrane of animal cell, as the animal cell will not have any cell wall. Okay, so and then the next one is the integral proteins. So integral proteins penetrate the hydrophobic core of the phospholipid bilayer. So you can put that in your note. Okay, hydrophobic core of what? Of the phospholipid bilayer. Okay, so as the phospholipid bilayer is the main structure for the plasma membrane. Okay, so integral proteins that span the membrane are called as transmembrane protein. So so you can either in your diagram, for example, if the question asks you to label, you can either label it as integral protein or transmembrane protein. Sama je. Okay, proteins that are embedded in the phospholipid bilayer of the membrane. Okay, so the hydrophobic region consists of one or more stretches of non-polar amino acid. So if you look at in this, okay, if you look at in this diagram, okay, so this is the transmembrane protein. Okay, this is the peripheral protein. It is located or attached at the surface of the membrane facing the cytoplasmic side, and you also have the cytoskeleton that will attach to the peripheral and also to the transmembrane protein. Okay, so this is to maintain the plasma membrane of animal cell. Okay, so for this protein, okay, the transmembrane protein, it has two regions. Okay, so the region, okay, that is in the core of the hydrophobic region of the membrane. Okay, so this is the hydrophobic part of the protein. So the one that is protruding out, okay, on this side and also on this side here. Okay, so these are the region of the protein that is hydrophilic. So it means that because this protein has both hydrophobic and hydrophilic region, so this protein is also amphiphatic. Okay, amphiphatic, same as the phospholipid molecule. So phospholipid molecule are also amphiphatic as it has both hydrophilic and hydrophobic region. Okay, so all right. So for the transmembrane protein, okay, so the region that is in the core, okay, which is the hydrophobic region of the protein, it is made up of alpha helical secondary structure. Okay, alpha helical secondary structure. So the hydrophilic region, okay, hydrophilic region of the protein that is protruding out of the membrane, okay, into the aqueous solution of cytoplasm or the extracellular fluid side here. So it consists of non-helical uh, polypeptide chain. Okay, non-helical polypeptide chain. So the 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 terms that you have to take note when you are describing the structure of protein, it says here alpha helical secondary structure of polypeptide. Okay, polypeptide chain. Bukan ya. Kalau letak is that alpha helical secondary structure of amino acid. Salah. Okay, amino acid is the monomer within the protein. So make sure awak tahu bila mana nak guna istilah-istilah ni. Alpha helical secondary structure of polypeptide chain. Okay, macam tu. So because it is consists of polypeptide chain, which is the monomer, is the amino acid. Okay, so the hydrophilic region, okay, non-helical, it is in contact with different aqueous solution. So outside, it is facing the extracellular fluid, okay, and then inside, you have the cytoplasmic side of the membrane, okay, within the cell. So okay, so these are the six major function of membrane protein. So first, it can function as transport protein. Yes, transport protein that are embedded, okay, which is the integral protein, embedded in the membrane and function as carrier protein, okay, carrier protein or channel protein. So you have two types, carrier protein and channel protein. For channel protein, it involves in the movement of ions or molecules across the membrane through passive transport. Okay, passive transport. So it means that from high concentration to low concentration of that ion or molecules. And then for carrier protein, it can involve in both, okay, passive and also active transport. Okay, so if active, for carrier protein that involve in active transport of substance across membrane, obviously it requires energy. Okay, the usage of energy. Okay, so second, okay, the protein can also function as enzymes. Okay, so the one example, okay, that I can give you is that tapi dia bukan dekat plasma membrane lah, kalau nak senang faham. Contohnya mitochondria. Okay, mitochondria will also have membrane. Dia apply sama lah. Cuma saya fikir sekarang ni adalah membrane of mitochondria. So dia juga membrane. Okay, so in the membrane of mitochondria, the inner membrane, it has enzymes. Okay, it has enzyme that function in the cellular respiration, which is to produce ATP. Okay, so far yang saya boleh bagi imagine untuk yang proses tu lah. Tapi there are many more enzyme that are found in the membrane, in the plasma membrane, and also in other membrane that have different enzymatic function. Okay, catalyze many different reaction. Right. Okay, tapi sebenarnya betul lah saya punya example tu. Okay, sebab tajuk dia pun membrane protein function. So it can apply for both. Okay, membrane of the plasma membrane and also membrane of organelles. Okay, so it can apply for both in this case. Okay, membrane of organelles and also plasma membrane. Okay, so yang the third one, okay, involved in signal transduction process. Okay, so this one is the one that I've been saying earlier, which is the protein, okay, which is embedded in the plasma membrane this time. Okay, plasma membrane, it has a binding site. Okay, it has a binding site for hormones to bind to. Okay, so certain hormone, okay, especially hormone that are made up of protein. Okay, so protein is a large molecule and it cannot easily pass through the membrane of cell. So in order for the hormone, awak tahu kan fungsi hormon adalah untuk kawal fungsi sel. Okay, untuk kawal fungsi sel terhadap sesuatu mekanisme. Okay, contohnya insulin. Okay, insulin is a hormone, okay, which is made up of protein, and the function of this insulin is to instruct the cell to convert glucose into glycogen. Okay, dia just mengarahkan, dia akan menghantarkan signal kepada sel untuk sel ambil glukos daripada luar sel masuk ke dalam sel dan tukar kepada glycogen. Macam tu. Itu fungsi hormon lah. So hormon yang dibuat daripada protein will bind to this membrane protein and triggers a series of reaction inside cell to catalyze or to involve in certain process inside of the cell. Okay, so the next one is involved in cell recognition. Okay, so yang ini, this one here is actually it involves the glycoprotein. Okay, the glycoprotein. That's why saya hari tu mention, okay, if you look at the structure of glycoprotein here, okay, so the glycoprotein will have a short chains of carbohydrate, we call it as oligosaccharide, attached to the protein. It becomes glycoprotein and it must face outside of the cell. Okay, so by having this glycoprotein, it can function as to identify, okay, this is cell of type 1. Okay, so this is cell of this tissue. Okay, so that is for cell recognition, to identify or to serve as cell marker. Okay, tu macam contohnya awak punya red blood cells. Okay, your red blood cell, how do you know that you are of type blood type A, blood type B, contohnya kan. So due to the presence of this glycoprotein that is present on the surface of the cell membrane. So maksudnya kalau darah A, darah B adalah antigen A, antigen B kan on the surface of the red blood cell. So this is due to the glycoproteins, okay, that are embedded in the membrane that forms the antigen, okay, of that red blood cells. Okay, so cell recognition contoh, okay, that is one example. Alright. Okay, so the next one is as intracellular joining molecule. Okay, so obviously, so for example, you have cells, okay, cells of certain type of tissues. So this cell has to attach to one another. Macam kita tengok hari tu tight junction contohnya kan, tight junctions. So this tight junction or desmosome will attach cells of the same type to form tissues. Okay, so this is involved in intracellular joining. Okay, intracellular joining molecule. And then the last one, it is to attach the membrane protein to attach to cytoskeleton and also the extracellular matrix. So if you look at this, okay, so this membrane protein, as you can see, it attached to the fibronectin here, kan? Fibronectin attached to the collagen, kan? Okay, macam tu. And then on the inside, it is attached to the cytoskeleton. Okay, so this is to maintain the shape of the membrane structure. Okay, so especially this one is only applied to animal cell. Okay, so as animal cell does not have any cell wall, so it has the protein has attached to ECM and also the cytoskeleton inside of the cell. Okay, so so this is the diagram, okay, to illustrate, okay, to you the function for each role of the protein. Okay, so as transport, so this one you can have channel protein. Okay, so another example, one example of channel protein yang common is aquaporine. Okay, aquaporine, which is channel protein that allows water molecule to diffuse, okay, to be transport through facilitated diffusion. Okay, facilitated diffusion from high concentration to low concentration. Okay, from outside to inside of the cell, for example. Water molecule, okay, without the presence of aquaporine, can still diffuse through the membrane for water molecule, but the diffusion of water molecule would be slower. Okay, will be slower. To increase or to speed up, okay, the movement, okay, of water molecule from outside into cell is by having these aquaporins. Okay, so kalau kita tengok chapter yang kita akan belajar sem depan, insyaAllah, on chapter homeostasis, you will be learning on how urine is formed in the kidney with the presence of nephrons. Okay, so nephrons at the distal tubule, kalau tengok balik, bukan, loop of Henle, okay, loop of Henle will have many aquaporins. Okay, it has many aquaporins to increase reabsorption of water. Okay, reabsorption of water in the kidney. Okay, so the next one here, you have the carrier protein. So this carrier protein, it can function in both active and also passive transport. Okay, so for active, okay, active transport, it requires energy from ATP. So it involves the movement of solute from low concentration to high concentration. Okay, so second, the protein can function as enzyme. Okay, so obviously because it functions as enzyme, it has active site. Okay, active site to bind for substrate to bind to the active site, and the enzyme will catalyze the reaction to produce products, and then this product can be the substrate to another enzyme. Okay, and then you have the actual product that you want. Okay, so this one is, for example, it involves a series of chemical reaction that occurs in the cell that involve several enzymes. Okay, so yang ini sebenarnya dalam apa punya sel ni akan melibatkan banyak enzim. Okay, banyak enzim that catalyze a series of reaction. Okay, so dia bukannya satu enzim you want one product only, kan? So your cell is really complex. It catalyzes many reaction that involve many enzyme. Okay, so the third protein, okay, is here, which is protein that has specific binding site that binds to a specific signaling molecule. Okay, so this signaling molecule, it can be hormones. Okay, hormones that are made up of protein. There are another type or group of hormones that is made up, or its component is from cholesterol. Okay, okay, cholesterol such as sex hormone, estrogen, progesterone, testosterone. So those hormones will not need to bind to this protein. Okay, they just can diffuse. Okay, for these hormones, sex hormones, steroid hormones, they can just diffuse across the plasma membrane to enter into the cell. But for hormones that is made up of protein, they cannot just simply diffuse through, because it is the protein is not soluble in the phospholipid bilayer. So they has to bind to this receptor protein. Okay, it has to bind to this receptor protein, and the binding of this hormone to this receptor protein, okay, will cause this protein to change shape. Okay, to change shape. So once the protein changes shapes, it binds to another protein inside of the cell. Okay, so this protein inside of the cell, it can be enzyme, for example, and this enzyme will catalyze a reaction, okay, or a series of reaction. We call it as signal transduction. Okay, a series of reaction inside the cell, to involve in certain reactions. Okay, certain reaction inside cell. So that is for signal transduction. And then D is cell recognition. So this one, it involves the glycoproteins. Okay, so this one, it can serve as cell identification. Okay, that cell identification of cell type. Okay, to recognize, oh, this is cell of type A, this is cell for type B. Okay, this is for cell for tissue A, tissue B, whatever. Okay, that is cell recognition. And then intracellular joining, macam kita tengok desmosome, tight junction. So this is intercellular joining. And then the last one is to attach, okay, to the cytoskeleton on the cytoplasmic side, and also extracellular matrix on the outside of the cell. Okay, so tengok istilah ni, extracellular matrix is the component that is consists of collagen, fibronectin and proteoglycan. Okay, the region outside, we call it as extracellular fluid. Okay, so ingat dua benda yang berbeza tu. Extracellular matrix is this one, collagen, fibronectin, okay, and proteoglycan. The fluid outside is extracellular fluid. Okay, so basically the next slide just to explain the whole protein. Okay, yang saya dah explain dah sama je. Okay, as transport protein, okay, it involve this protein, carrier protein. Okay, so the plasma membrane or cell membrane are very, very selective, allowing only certain solute to enter or leave the cell, either through channels or carrier composed of proteins. Okay, so you have to type lah, kan? So channel protein and also carrier proteins. Okay, so for channel protein here, or we can also call it as pore protein. They have a region at the center here, which is, we call it as hydrophilic channel. Okay, hydrophilic channel. Why do we call it as hydrophilic channel? Because it allows ions, for example, okay, ions to pass through the protein, get through this channel to enter into the cell or to exit the cell. Okay, sebab tadi dia cakap enter or exit. Okay, enter or leave. So that is selective for particular solute. So certain ions, okay, will have their own type of channel protein. Macam contohlah saya bagi tadi kan, kalau air, the channel that involve in the movement of water, we call it as aquaporins. Okay, dia tak boleh lah awak guna aquaporin tu untuk bawa masuk chloride ion or sodium ion. Okay, so every ions or small polar molecules, okay, such as water, will have their own channel protein. Okay, and the next to it, the protein that is on this side here, you have the carrier protein. Okay, so carrier protein will have binding site. Okay, binding site for the solute that the protein wants to transport. Okay, so when the solute binds to this binding site of the carrier protein, it causes the carrier protein to change shape, okay, to change shape. And therefore, when the protein changes shape, the solute can be transported across the cell. So in this case, the carrier protein involve the movement of solute from low concentration to high concentration. So this is a type of active transport that requires energy from ATP. Okay, so highlight kat sini kalau untuk carrier protein, it involve the protein to change shape to move solute, okay, across the membrane. Okay, so the next one here is involved in enzymatic activity. Okay, so again, for this protein that function as enzyme, the active site, okay, must face the cytoplasmic site inside the cell, because the reaction obviously has to occur inside cell. Tak bolehlah berlaku kat luar kan. Okay, it has to occur inside cell. So please take note that the active site will face the cytoplasmic site to catalyze the reaction inside the cell. Okay. So a protein built, okay, into the membrane may be an enzyme. Okay, it function as enzyme. It has active site exposed to substances in adjacent solution. So several enzymes organized to carry out sequential step of a metabolic pathway. So a metabolic pathway may involve a series of reaction that involves several enzymes. Okay, so that's why in this picture you can see it illustrates two enzymes that catalyze two reactions. So this is just to make you understand lah that inside cell you have metabolic pathway that actually involve many reactions that is catalyzed by several or many enzymes. Okay. Okay, so the next one, the third one is the protein can function in signal transduction process. Okay, it says here membrane protein acts as a receptor. Okay, so a receptor, why is it act as a receptor? Because it has specific binding site here, okay, for the signaling molecule to bind to. Okay, has a binding site with a specific shape that fits the shape of a chemical messenger. Okay, chemical messenger or signaling molecule in this case, for example, is the protein. Okay, is the hormones. Okay, hormones that is made up of protein. So the signaling molecule causes the protein to change shape. Okay, to change shape. So in this case, for example, asalnya, on tengok asalnya, kalau hormon tu tak bind, okay, shape dia macam ni je. Okay, shape dia macam ni, and inside inside the cell another protein, which this one can be enzyme. Okay. Okay, dia tak adalah macam substrate or substrate ke untuk bind kan, ataupun component to activate the enzyme, for example. Okay, so once this signaling molecule bind to the receptor protein, it causes the protein to change shape. Okay, so this change in shape fits, okay, the enzyme here and causes the enzyme to be activated, for example. Okay, so this enzyme then can catalyze certain reaction and then involve another enzyme, and then another enzyme catalyzes another reaction within the cell. So this is what we call it as signal transduction. It relays, okay, it relays, okay, message inside the cell. Okay, so this one is for signal transduction. Okay, so the next one is involved in cell recognition. Okay, cell recognition. So cell recognition, which is a cell ability, yeah, a cell's ability to distinguish, okay, untuk kenal pasti one cell or one type of neighboring cell from another. Okay, so membrane carbohydrates may be covalently bonded to lipids, which when the carbohydrate binds to lipid, we call it as glycolipid, or the carbohydrate can bind to proteins. Okay, in this case is the protein here to form the glycoprotein. Okay, so remember the binding of this carbohydrate must be on the outside of it faces outside of the cell. Okay, it can attach to this phospholipid to form the glycolipid, or the carbohydrate can attach to protein called as glycoprotein. So this is to allow the cell to identify itself, okay, or identify the neighboring cell. Okay, so some glycoprotein serves as identification tag. Okay, identification tags macam saya cakap tadi lah, antigen pada red blood cells. Okay, that determines, oh, this is blood type A, this is blood type B. Okay, for red blood cells of that does not have any antigen on the surface, okay, untuk orang tu dia adalah blood type O, because for people that has no antigen on the surface of red blood cell, those people are blood type O. Okay, the red blood surface does not have any antigen. So that's why untuk orang yang berdarah O, sebab dia tak ada antigen kat atas ni, dia tak ada carbohydrate, okay, binding to the protein. So that's why for orang yang berdarah O, dia boleh terima darah daripada orang darah A, darah AB, darah B. Sebab apa? Bukan, dia tak boleh terima. Sekejap. Dia boleh derma. Kan? So salah saya terbalik. Orang yang berdarah O boleh derma darah kepada sesiapa je pun. Okay, orang yang berdarah O boleh derma darah kepada sesiapa sahaja, darah A, darah B, darah AB, darah O. Sebab apa? Bila darah orang yang berdarah O tu masuk ke dalam penerima dia, okay, penerima dia, penerima punya antibody takkan attack. Okay, takkan attack these red blood cells that enters into the recipient body. Okay, recipient body, kalau dia terima foreign substance, dia akan ada antibody yang akan attack. Okay, orang darah O sebab dia tak ada antigen, so antibody penerima tu takkan attack. Macam tu. Okay, faham ke? Faham tak? Ke tak faham?
Faham.
Okay. Alright. Alright. So tu contohlah. So yang pasal darah-darah ni kita akan belajar masa semester 2. Chapter mana? Chapter immune tak silap saya. Okay. Alright. Okay. So some glycoprotein serves as identification tags. Okay. So ini contoh yang saya bagi tu, dia dah bagi tahu. Okay, so cells recognize each other by binding to surface molecule, often carbohydrates or protein on the plasma membrane. So as importance, sorting of cells into tissues and organs in an embryo. Okay, so this is to make sure that cells of the same type assembled together to form functional tissues, and tissues form functional organs. Okay. And then second, basis for the rejection of foreign cells by the immune system in vertebrate animal. Okay, so sometimes basis for rejection of foreign cells by immune system in, okay, macam saya cakap tadi lah, kalau foreign substance, pathogen, okay, substance that is not supposed to be in your body. Okay, so your immune system will attack this foreign cell because this foreign cell will have the glycoprotein that will become the antigen, okay, for the antibody of the host to attack. Okay, so host badan awak tiba-tiba masuk foreign substance. So this foreign cell, okay, so this foreign cell will have antigen on the cell surface. Your body will produce antibody that will attack that foreign cell. Okay, so that is the basic for the rejection of foreign cell. Okay, so if I were to draw contohnya, okay, a virus contohnya. Okay, virus. Okay, so virus dekat surface dia akan ada antigen. Okay, so this antigen is from the carbohydrates that bind to the protein, kan? So that forms the antigen. So when the antigen enters into the host body. So the host will produce antibody, will produce antibody that will attack the antigen on the surface of the pathogen. So bila masuk, the antibody binds. The antibody binds, and this can cause the foreign substance, in this case, for example, the virus or pathogen, to be inactive. Inactive. Tak boleh nak masuk dalam sel-sel badan awak untuk merosakkan sel badan. Will be inactive. So once this pathogen is in the inactive state, so then will come another white blood cell. We call it as macrophage. Macrophage. So this macrophage will engulf the virus or the pathogen or other foreign cell that enters into your body, engulf and then destroys. Engulf through phagocytosis lah. So itu contoh yang mudah. Untuk fahamkan basis for this rejection of foreign cell by the immune system. So the third one is carbohydrates on the external side of the plasma membrane vary berlainan, among species, individuals, and cell types in an individual. So contohnya, that's why kalau macam nak pindah organ, nak buat organ donation, kena check darah semua supaya orang yang darah sama je boleh derma contohnya lah ataupun the antigen surface, the antigen on the cell of the donor is the same as the recipient, so that the recipient body will not reject that the organ that is being donated tu. So that's why basically species cells of species will be different, species will be different from another species individuals from another individual, dia punya antigen at the cell surface will be different. Will be different even cell types in an individual. So example is the human blood type, which is A, B, and O. So blood cell, and red blood cell So kalau O tak ada antigen. Kalau B contohnya antigen dia macam ni. Kalau A contohnya macam ni, dia punya antigen dia, contohlah. Kalau AB antigen dia dua-dua ada. Yang tiga segi, yang bulat macam ni. Yang tiga segi. So that's why AB. So detail pemahaman pasal blood type ni kita akan belajar masa chapter genetik dan juga immune. During semester 2. So the next one is the function of the membrane protein as intercellular joining molecule. So membrane proteins of adjacent cells may hook together in various kinds of junction, such as gap junction and tight junction. So this one we have looked at during chapter 2, which is the function of gap junction is to provide cytoplasmic channel between adjacent cells so that substance from one cell to another cell can be transferred directly through the cytoplasm that is connected between the cell. And then tight junction. Tight junction to prevent leakage of extracellular fluid. So this is, you have one cell, and then you have another cell, and then you have another cell. So you have the presence of this tight junction that attach these cells together and prevent extracellular fluid from leaking in between this cell. So that is the function of tight junction. So next, attached to the cytoskeleton and extracellular matrix. So it says here microfilaments on the inside of the cell here, microfilaments and other elements of the cytoskeleton may be non-covalently bound to the membrane proteins, okay, like this one here and this one here. So the protein will attach to the cytoskeleton, which is to maintain the cell shape. Especially this one only applies to animal cells, okay. Animal cell, it applies to animal cell, okay. It stabilizes the location of certain membrane protein. So dia juga menstabilkan protein tu daripada bergerak terlebih, kan. So bergerak terlebih. So this is to stabilize the location of the membrane protein. The summative question, this one you can do on your own. Name two types of transport protein. So senang je, apa dia? Sender protein and carrier protein. So second, briefly describe how membrane protein involved in signal transduction. This one you have to explain yang hormone tadi kan, contoh signaling molecule, chemical messenger that binds to the receptor on the on this protein and causes the protein to change shape and release information inside the cell. Okay, it causes a series of reaction to occurs inside cell. We call it as signal transduction. Okay. Okay. So next kita maybe boleh pergi lagi. Okay. Okay. So membrane structure results in selective permeability. Saya rasa kita kena stop lah dulu kot sebab saya lupa. S48 you have tutorial next kan? So we stop here for now. Okay. So we are going to continue for 3.4 next week. Okay. So thank you class. Kalau S48 you may leave this session. S47 you may maintain. Okay. Thank you.
Thank you, madam.
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 Okay. Kalau ada nak tanya soalan boleh. Saya lupa nak tanya if you all have question, but if you do have, you can still ask. Mana yang tinggal ni?